So I'm going to, first of all, I'm going to be walking around a little bit because I find it easier to point from here where I can actually see the slides. I hope that doesn't distract you all. And I'm going to be talking about therapeutic drug development in general. So how do we approach developing treatments and ultimately a cure for rare disorders like Smith-Lemli-Opitz syndrome? And then I will also tell you more towards the end of the talk a little bit about work that we're doing, very early stage development of drug therapy, hopefully for Smith-Lemli-Opitz syndrome. All right, so first a little bit about orphan disease background, rare disease background, and some statistics. There'll be a lot of statistics in the first few slides. I also want to say I do want to leave time at the end for questions, so please do feel free to save your questions for afterwards. So rare diseases by the numbers. I think some of these numbers will really surprise many of you. So rare diseases affect 30 million Americans. That's a lot of people. That's 1 in 10 of us. There are approximately 7,000 different rare diseases that exist. There are nearly 500 orphan drugs already, and 1/3 of all new drug approvals are for rare diseases, really a startling statistic. Now, 80% of rare diseases are genetic, so not all rare diseases are genetic, but most of them are. And then there are still many different diseases for which we do not have therapeutics, but there are 450 medicines currently in development for rare diseases. So we are making progress, but that progress is slow. The Orphan Drug Act from 1983 has been incredibly instrumental in our development of therapeutics for rare diseases. If you think about it, if there's a disorder that only affects a few hundred people in the United States, a pharmaceutical company doesn't have a big incentive to develop treatment for that disorder because it's terribly expensive to develop therapeutics, and they're not going to recoup their investment by treating only a few hundred people unless there are specific incentives. They're much more incentivized to develop treatments for common disorders because they'll make higher profits. The Orphan Drug Act of 1983 is a federal law that incentivizes biopharmaceutical companies to develop drugs and biologics known as orphan drugs for individuals with rare diseases. And a rare disease in this case is defined as a condition that affects less than 200,000 individuals in the US. Now, 200,000 doesn't sound like a rare disease, and it's true, most of our rare diseases that we typically think about are much less common than that, and SLOS is in that category. There are much fewer than 200,000 Americans, but that's the cutoff that's used. What are the incentives that are given in the Orphan Drug Act to the pharmaceutical companies? Well, there's 7 years of marketing exclusivity. So this is huge. When a drug company develops a treatment for a rare disease and they get FDA approval, no other therapeutic will be allowed on the market to compete with that drug for 7 years. They also get a 25% tax credit for qualified clinical testing expenses. There's user fees that companies pay for the FDA to review their drug for approval. That is typically around $2.5 million, and that user fee is waived when the drug is for a rare disease. And finally, this is a more general, this doesn't go to a specific pharmaceutical company, but there are millions of dollars in research grant funding to assist with drug development for rare diseases. So it's really been a remarkable achievement. Has the Orphan Drug Act worked? I think many of us would, or most of us, almost all of us would say yes. Now, I will say there are some critics of the Orphan Drug Act, so there are people that are challenging this because there are companies that have benefited tremendously for this. So there are companies that have developed drugs initially for rare diseases. The drugs have been approved, they got in marketing exclusivity in those 7 years, and then later on, that same drug will be used for more common disorders. And so there are companies that have made billions of dollars. Here's some help from Juki. The companies, do you ever feel like somebody's looking over your shoulder? There are companies that have made billions of dollars in profits on drugs that initially were developed for rare diseases. But I think you could argue, good for them. Initially, they did develop the drug for the rare disease. The drug has worked, and everyone has benefited. But look at the numbers here. The number of approved orphan indications has grown tremendously from about 34 in the initial years of the Orphan Drug Act to more than 730. But still, about 95% of rare diseases do not have a specific FDA-approved therapy. All right, now into more of the specifics of the drug development process itself. And some of you are familiar with this, others of you may have little to no familiarity, so I'll go slowly here, and I have a few slides I think that illustrate this. But when drug companies or academicians are looking to develop treatments, they may initially do what's called high throughput screening. They may look at about 10 to 15,000 different compounds to find a drug. And out of those, there may be about 250 of those compounds that look promising initially. 5 of those have features that make them potentially safe for testing. And in the end, only 1 of those compounds is typically approved as a marketed drug. So 1 out of 10 to 15,000. And look at the time it takes to take a drug from initial concept to approval. The early stages, 3 to 6 years. The clinical trials where you're testing the drug in humans, 6 to 7 years. And then the FDA review and the manufacturing process and post-approval, 6 months to 2 years. So really, the duration from taking a drug from initial concept to approval and availability can be 10 to 15 years, and the average cost can be over $1 billion. Now, this is for drugs in general. There are some ways we can shorten that and reduce the cost for orphan or rare diseases, but for common disorders, these are the statistics here. So the early phases, pre-discovery, drug discovery, preclinical work, this means research in the test tube and in animals. Then the clinical trials where you're testing the drug in humans, and there are phase 1, 2, and 3 clinical trials. And then, as I said, FDA approval and post-approval, there are often required phase 4 clinical trials or phase 4 studies that I'll tell you a little bit more about in a minute. All right, so back to the Orphan Drug Act. As I said, taking a drug to market typically can cost billions of dollars. But rare disease patient advocates and legislators worked with the FDA to create the Orphan Drug Act to encourage pharma to develop drugs for rare diseases which otherwise might not be profitable. Orphan drugs are those for whom the number of patients with the disease is smaller. And just to reiterate, the cutoff is typically 200,000 persons in the US. In reality, most diseases are far less prevalent than that. So the Orphan Drug Act gives companies who develop these rare therapeutics for rare disorders pathways to shorten or simplify the development timeline and provides them with longer-term protection to economically recapture the costs of development, including things like tax incentives. The early stage development of drugs is often not very visible. It's very much behind the scenes and hard to even find out about sometimes. And this is where these 10 to 15,000 compounds are screened and compounds are identified that may look promising. So typically this is highly scientific work carried out by scientists dedicated to research in academia or pharma. It's theoretical research based on concepts trying to understand the disease. It's proof of concept testing. It's testing in what we call in vitro, in the test tubes or in animals. And importantly, the way that pharmaceutical companies make a profit is by having intellectual property, which is typically in the form of patents. This protects them from competition and allows them to make a profit once a drug is marketed. The approach for rare disease is typically more opportunistic, so often it leverages off of findings from research in other areas, maybe common disease areas, And it's also futuristic, demonstrating something that can be useful for a larger disease area in the future. The goal initially is to screen ideas to test in patients. Can we model a disease? Sometimes the models are in cells in the test tube, sometimes they're in animals. Does the drug perform as expected? Does it cause harm? Is it toxic? Does it have side effects? Funding for all of this is critical. We were talking earlier, sometimes we think that this is all that we do is about money. But really, if you don't have funding to do the research to develop therapies, there's no place to go. Will the studies and the concept be strong enough to convince your funders? So the funders initially, to allow research to go forward, include grants from institutions like the National Institutes of Health, National Science Foundation, and so forth. Angel investors, these are really early investors. Universities, venture capitalists, the patent office is important here, and the technology transfer offices at our universities, and pharmaceutical industry as well. All right, so we have an idea, we have a drug that we want to develop, we do the initial preclinical test tube studies, animal studies, and now we want to move to humans. Testing in humans is done very cautiously. We have to make sure we're doing everything we can to protect the patients from being harmed by the therapeutics that we're developing. So before the research can go forward in humans, it has to be run past an institutional review board. Often in other countries they're called ethics boards. And these IRBs are in hospitals or institutions. They review the research to make sure that it seems like it's very likely to be safe and potentially has some therapeutic potential. The justification for using the drug has to be compelling. These drugs also have to be approved by the FDA to be used in humans as well. So we have to submit what's called an investigational new drug application, or IND, that the FDA has 30 days to review. I alluded to the phases of testing drugs in humans, and you'll see this again and again in the talk today. Typically, the phases we're thinking about in clinical trials are phases 1 through 3. Phase 1 is initial in human studies testing the safety of a drug. Phase 2 is looking at the effectiveness at treating the disease, and we do things like look at different doses in phase 2 studies. And then phase 3 is larger scale safety and effectiveness studies, really the studies that form the basis of a submission to the FDA looking for marketing approval of the drug. Phase 4 studies are often required, especially in rare disease studies, where there are fewer phase 1 to 3 patients that have enrolled. And so the FDA is interested in gaining as much data as it can long-term about safety. The standard process, this is not in rare diseases, but in more common diseases, typically we have about 20 to 100 typically healthy volunteers in a phase 1 clinical trial, seeing if a drug is safe. Then we have subpopulations that are tested, 100 to 500 or even 1,000 to 5,000 people in phase 3 studies. You can imagine trying to do that in a rare disease would be impossible. And these studies can take 6 to 8 years. At the end of this, there's a submission to the Food and Drug Administration for marketing approval, typically after the phase 3 studies. What about rare diseases? There are challenges in rare diseases as well as opportunities. Some of the challenges are the small populations, the small numbers of affected patients available to even enroll in the clinical trials. Often there are few treating physicians and treatment centers. I mean, look at how few of us there are in the SLOS community who are seeing these patients. The disorders are often highly heterogeneous. The patients are affected differently. Some are really severely affected, some are more mildly affected. There's a wide range of severity and a difference in the rate of progression of the disease in some cases. Often rare diseases are less well understood, and their natural history, in other words, what signs and symptoms and complications of the condition do the patients have, that varies and is incompletely described. So we know a lot about heart disease. We know a lot about hypercholesterolemia because there's so many people affected, and they've been studied in very large studies. But for rare disorders like Smith-Lemli-Opitz syndrome, there's still a lot to learn. Most rare disorders are serious or life-threatening. There are significant unmet medical needs, and there's a lack of regulatory or drug development precedent. So for things like Smith-Lemli-Opitz syndrome, there haven't been a lot of clinical trials already done. We don't know what outcomes to measure. In other words, if we develop a drug that's a therapeutic for a rare disorder, we may not know what to even look for in terms of improvements to be expected because we don't know the natural history very well. And we may not have validated means for measuring the outcomes. So when we're developing a drug for hypertension, we measure blood pressure. Well, what would you measure in someone with Smith-Lemli-Opitz syndrome to see if a therapeutic is helping? And finally, rare disorders, many of them affect children. The patients are predominantly pediatric. That provides a whole new set of challenges because there are things that we can do in adults when we're studying diseases that we're not allowed to do in children. All right, so what's been done in terms of rare diseases to facilitate development of the drugs and really incentivize is the FDA has allowed for modification of the clinical trial requirements. So the standard process I already went through here, the phase 1 through 3 studies you can see over here. In rare diseases, the phase 1 is often done in affected patients, not healthy normal volunteers. The size of the studies are much smaller. Often these are like 10 to 12 patients in a phase 1 clinical trial. And sometimes the phase 1 and 2, the safety and effectiveness studies are combined in rare diseases. So there are a few examples where pharmaceutical companies have sought and even received FDA approval after single combined phase 1 and 2 clinical trials. That's often for ultra-rare disorders. And then if phase 3 studies are required, they may not need to be randomized treatment versus no treatment. Rather, we may look at low dose versus high dose, or we may look at treatment versus natural history, untreated patients. So there's some flexibility that's introduced in the system by the Food and Drug Administration to allow these studies to go forward. And then, as I said, phase 4 studies are often required post-approval. So rare disease FDA approval process can be smaller and faster. What are the odds of success taking a drug from concept to approval? So here we see the probability of success measured in percentages, the different phases of the studies, and then finally over here, phase 1 to approval. We'll get to that in a minute. The green is for rare diseases, the bluish color, the dark colors for all diseases, and the yellow is for chronic high prevalence, more common disorders. So I think what you can notice right away is that for all of these, the probability of success is actually higher in rare diseases. Phase 1 to phase 2, the odds are pretty good of being able to go from phase 1 to 2, about 75% in rare diseases. Phase 2 to 3, it drops, but there's a significant difference. Rare diseases often do go from phase 2 to phase 3. And then from phase 3 to a new drug application or a biologic license application, these are the applications to the FDA asking them to approve a drug. Those odds are about 75% in rare diseases, not bad. And then once you get to the point of an NDA or a BLA and you're submitting to the FDA, the odds are really very good. But getting to that point is tough. So going all the way from a phase 1 study to FDA marketing approval, about 10% chance in common disorders, 25% chance in rare disorders. All right, so now narrowing it down to what we're most interested in here, Smith-Lemli-Opitz syndrome. What are the current treatments available? And I won't go into this in any detail at all since you heard so well from Katie already, and you'll hear from others later in the conference. But oral cholesterol supplementation is often used, things like egg yolk, cholesterol suspensions, and commercially available cholesterol formulations. But so far, there's no solid evidence that cholesterol supplementation is an effective treatment. And feel free to ask me about that later, because some of you may have questions about that. Cholesterol-lowering drugs, these statins or HMG-CoA reductase inhibitors, have been tried based on animal and human cell studies showing that they increase the residual enzyme activity and reduce 7-dehydrocholesterol accumulation, but we can ask Dr. Porter about that later. So no specific treatment and no cure to date. What are our goals as we're trying to develop treatment for SLOS? Well, we want to increase brain cholesterol, and we want to decrease the 7-dehydrocholesterol and 8-dehydrocholesterol. At least I think that the condition is caused by both cholesterol deficiency and accumulation of the potentially toxic DHC precursors of cholesterol. We want to improve feeding and growth. We want to improve development, cognition, and IQ. We want to improve behavior, and we want to improve the autistic features that are often found. So the treatment ideas, we've gone through most of these. One thing we didn't talk about is highly concentrated intravenous forms of cholesterol. So we can concentrate cholesterol from things like fresh frozen plasma, or an idea that we've had, there are patients with very high cholesterol levels that have a procedure called apheresis that takes out their lipoprotein cholesterol, removes it, and then that's just discarded. Well, maybe we could save that and actually infuse that back into patients who have cholesterol deficiencies. Other therapeutic statins, we talked about antioxidants and bile acids. Cholesterol or enzyme delivery to the brain. We've got to get cholesterol across this blood-brain barrier. So when we have cholesterol in our blood, it doesn't get into our brain. Our brain makes its own cholesterol. So that's a challenge in SLOS. Can we implant stem cells in the brain, stem cells that have normal amounts of cholesterol? And can those propagate? Can they grow in the brain, stay in the brain, and move throughout the brain? Those kinds of studies have actually been done in other diseases. So we did one of those studies at Oregon Health and Science University in another disease where we implanted human fetal neural stem cells into the brains of children with neuronal ceroid lipofuscinosis. Unfortunately, we were only allowed to do a phase 1 clinical trial, so we don't really know whether that was effective or not, but more of those studies are coming. Prenatal treatment, high cholesterol diet, cholesterol delivery to the amniotic fluid or fetus is a possibility through the umbilical vein or into the peritoneum, into the abdominal cavity. And then organ, bone marrow transplant, and gene therapy. So a lot of these are ideas that haven't really been exploited very much yet in SLOS. So now I want to spend some time talking about some very early stage research that we are carrying out to try to develop a new treatment for SLOS. And this is our team. Our goal is to bring cholesterol to the brain to treat SLOS. We are the Neurodel collaboration. This is the team here. Caroline is here today. Caroline I will refer to as the matchmaker. She works with an accelerator program to bring scientist groups together to work on problems like treatment for SLOS. I have to give a lot of credit to Craig Smith here. He goes by Smitty. Smitty worked for Sanofi for a long time as a scientist. He came across some of our research in Smith-Lemli-Opitz and became fascinated by it and really taken by the stories of affected children and wanted to do something to help. So he actually contacted me, sought me out, and said, what can I do to help? And he's been a great help. He introduced me to Caroline. So our clinical need, as I said, is to deliver cholesterol to the brain. And Caroline introduced us to ZJ and Roman. So these are chemists who are developing compounds to get cholesterol into the brain and then release that cholesterol in the brain. And we owe a lot of gratitude to the SLOS Foundation for approving a grant to support our work. So the concept, increased cholesterol to the brain. The challenges, As I said, free cholesterol circulating in the blood cannot cross the blood-brain barrier. For SLOS patients, there's not enough cholesterol in the brain. So we're developing compounds to get into the brain and deliver cholesterol. So these would be delivered initially probably by injection, but later potentially even by mouth, travel across the blood-brain barrier, increase brain-based cholesterol, and then cholesterol is available for cellular interaction in the brain. The specific aims of our project, initial proof of concept and drug modification as needed, so it would ultimately be able to be used in humans. Increase cholesterol, quantify the degree to which a therapeutic increases cholesterol in the brain. Specific achievement of this aim would allow the ability of the therapeutic agent to increase cholesterol in an environment simulating the brain. Therapeutic delivery across the blood-brain barrier. In mice, we would determine the accumulation of cholesterol in brain tissue following administration of our lead compound. And successful achievement of this aim would indicate the ability of the compound to successfully reach the brain following systemic delivery to the blood or to the mouth and increase brain cholesterol. So we've actually developed a couple of compounds, and our lead compound underwent early testing. It was put into a test tube that had chemicals simulating the brain chemistry. This slide, so these are two of the actual results slides, looking at concentration of cholesterol on the y-axis and time on the x-axis, time in minutes. And you can see that there was an increase in cholesterol over time in this test tube simulating brain chemistry from over 0 to 4 hours. This slide, or this figure shows the percentage of cleavage of the compound and what percentage of the compound was used to increase cholesterol, and you can see that it's over 50%. So under in vitro cell-free conditions, well over half of the compound was used to increase cholesterol, and we could measure that increase. So again, very early results. We still have a long ways to go. We're about here in the status of drug development. So we had a hypothesis. We developed a chemical, synthesized the chemical, 2 chemicals. Proof of concept, you saw our test tube studies. We're increasing brain-based cholesterol in a simulation study. We still have to study stability and reproducibility. And then ultimately, we want to test this in mice. We will have to optimize the drug or drugs that we develop. There are studies called ADME, absorption, distribution, metabolism, and elimination. We have to learn as much as we can about how the therapeutic will react in a human. And then safety and toxicity studies, the phase 1 clinical study based on all of this, what we call preclinical work done before the drug is even used for the first time in people. All right, and we hope to report back in the future as we have more results. So, other current SLOS research, I just want to end by telling you a little bit about other things that are going on and how you can find out about the most current research in SLOS. So as Katie alluded to, this website, clinicaltrials.gov, is a great place to look. If you search for Smith-Lemli-Opitz syndrome, there are 14 studies. Some of you may think that's a lot, some of you may think that's very little. In the end, though, in terms of studies that are actually active, there's one that's not yet recruiting, but is, I think, considered active, and Ellen can tell you more about this, Smith-Lemli-Opitz syndrome and cholic acid. This looks like it'll be done at Colorado Children's, University of Nebraska, Cincinnati Children's, and Children's Hospital of Pittsburgh. Those are at least the potential sites. There are 2 studies that are currently active and recruiting patients. In other words, you could sign up to be in these studies right now. One is cholesterol and antioxidants at Children's Hospital of Colorado with Dr. Ellen Elias, and the other is with Dr. Denny Porter at the NIH, a natural history study, learning as much as we can about SLOS so we can develop these outcome measures and be able to tell whether a therapeutic is actually even going to help. Pharmaceutical companies are starting to show interest in SLOS. This is new, we hadn't seen this before. So there's actually, I found a website for Sage Therapeutics, and they're talking about development of cerebrosterol for Smith-Lemli-Opitz syndrome. We still need more SLOS research. This slide shows the number of publications, the number of articles that people have written about Smith-Lemli-Opitz syndrome over the years, starting in 1964 with the first report, ending this slide, I think I made it in about 2016 or 17. And then the defect in cholesterol synthesis described by Mira Irons, Ellen Elias, Tint, and Saline was in 1993, so there was a spike in the number of articles, and the number of articles has continued much higher than in the early days. Then in 1998, the 3 groups reporting on the identification of the gene responsible for Smith-Lemli-Opitz syndrome in 1998. But I think many of us were hoping to see a continued spike in the number of articles. That really hasn't happened very much. When you search Smith-Lemli-Opitz syndrome and treatment, there are only 38 articles. And when you limit that to humans, only 26 articles. So I don't think I have to convince all of you there's a great need for research in Smith-Lemli-Opitz syndrome. So my last slide, the importance of research funding, I alluded to this earlier. There are lots of challenges in Smith-Lemli-Opitz syndrome, a small number of people, diverse presentation. Animal models are not great. The physiology is challenging. We have to get cholesterol into the brain. The needs in research, we still need better basic understanding. We need better efforts towards delivery of cholesterol or other therapeutics and better animal models. Our goals are pretty simple: find treatments and cures, improve the lives of the patients, and funding from advocacy groups like yours is absolutely critical. So I'll stop there and be happy to try to answer your questions. Thank you.
Do we have questions for Dr. Steiner?
Hey, sir, how are you? Can you hear me?
Yes.
I just had a quick question. You said the Orphan Drug Act, the requirements are sometimes reduced.
Yes.
Okay, so say a company takes it 7 years later, I could just go like that. 7 years later, another company could come take that drug and then say repurpose it for other conditions.
Or develop a generic that's actually the same thing and sell that, yes.
In those situations, do they have to then take that drug and do their own phases of trials, or do they just kind of piggyback off of what was previously done?
Yeah,
Second
great.
part to that is,
Yeah.
sorry, before I forget, do they ever feed back that information to the original researchers or the original drug developers to kind of better enhance, ongoing?
Right, so after the 7 years of exclusivity and no competition, another company can come in, develop essentially the same drug, a generic, if you will, and they have to still submit to the FDA, but if they're using the same compound, you can imagine that the submission and the clinical trials are much simpler. Basically, all they have to do is show that their drug is the same, and then they can refer to all the other information that the first drug had on record with the FDA. So much simpler process, much quicker, fewer clinical trials, maybe one clinical trial, which is called bioavailability. That may be all that's necessary. Do they ever feed back? So what you're talking about is a second company coming in, developing the same drug, but let's say it's a generic, it's going to be lower priced, it's going to be competition. There's no incentive for them to give more information back to the first company.
Cool. Thanks.
Mm-hmm.
Other questions?
Yeah, we've got a fight for questions over here. Thank you, Dr. Steiner.
Sure.
So, regarding the exclusivity clause, so I understand from a pharma perspective, there's some incentive there to have that exclusivity, but from a clinical perspective, why have that, and don't we limit our ability then to get these things into the clinical world, get these drugs potentially into the clinical world?
Yeah, great question again. It is a trade-off. So as a community of rare disease advocates, patients, and doctors and researchers working together on the disease, I think you'd love to have as many different potential therapeutics as possible. And this period of marketing exclusivity can actually prevent additional therapeutics from coming in. In some ways, that's a good problem to have. It means that there already is one therapeutic, there already is one drug. I think we would never get to even one drug without the Orphan Drug Act. So at least you're getting something. But you're right, it's not a perfect system because it may be preventing some other therapeutics from coming onto the market.
Yeah, I mean, I can imagine that there might be some subtle differences between drugs that would get around that exclusivity.
That's exactly right. Oftentimes, if there is a second therapeutic, it's different enough from the first one that it can also be approved.
Yeah.
But if they really are the same concept, only one can be approved.
So my other question is, so you mentioned the statins, you know, the idea of using that for treatment. Are there other potential drugs that we're chasing down that could be repurposed that are already FDA approved that we could repurpose then for treatment of whatever the symptom might be within the SLOS?
I'm a big proponent of repurposing drugs that we already know a lot about safety and toxicity. I don't know that researchers in this field have identified other drugs, though, that could be repurposed for SLOS because it's such a kind of fairly unique situation. But if there are other drugs that are already approved or have been studied, could be repurposed for SLOS, I think we're all supportive of that.
Yeah. And is there another disorder, rare but more commonly known, that's dealing with this low cholesterol that they're also working on the cholesterol problem? I know high cholesterol, you mentioned.
Yeah. All of the, let's see if, I don't think this is an overstatement, all of the low cholesterol disorders are rare. SLOS is probably, in the group of cholesterol synthesis disorders, where you have low cholesterol because you're not making enough, it's the most common of those disorders by far. The other disorders have 10 or 12 patients in the world ever described. So I think there the efforts are predominantly in SLOS. There are some other disorders, different gene defects that have low cholesterol, but there it's not intracellular low cholesterol. So those are different disorders. I think the treatments are going to be different. And for those disorders, which may be a little more common than SLOS, there aren't very good treatments for those either. But the symptoms of the diseases are not as severe as SLOS, so there's not the same incentives to develop expensive, potentially toxic treatment. So the short answer is, it's a really great question. If we could learn from other more common disorders with low cholesterol and use that information to treat SLOS, I don't think that applies very well here.
Okay, thank you.
Are there any other studies going on elsewhere in the world? And do you communicate with each other? Because we're aware of some study that's been done by Professor Clayton at Great Ormond Street Hospital in London in the past. Is there anything happening now?
Right. So
not necessarily just in the UK, though.
Yep. So clinicaltrials.gov, that website is really international. I don't know about the regulations in Europe, whether they have to report to clinicaltrials.gov like we do in the US. But by and large, if there's a clinical trial, a therapeutic clinical trial in SLOS, it will be listed on clinicaltrials.gov. So the short answer is there's not a lot going on currently in the rest of the world for treatment studies for SLOS. We do communicate with our international colleagues. I think Dr. Porter and I know Dr. Clayton, Professor Clayton, and these other people that have been involved in SLOS research. We have international meetings. We try to attend those meetings that the Europeans and the Japanese and others attend as well. So there is crosstalk there. It can always be better, you know, if there were. So maybe you've stumbled on a great idea. You know, maybe the next. Maybe in the future, these meetings could be even more international in scope, and maybe we could have one in the UK and the next year in the US. Anyway, it's a great concept to be talking about expanding the research and the research outreach to be truly more international.
My question is more based on your research that you're doing. Is the hope that this drug will be helpful for those of us that have older children? Will we get benefit from crossing the blood-brain barrier with the older kids?
Right. I think there is hope for improvement even in the older kids up to a point. So I don't think there's a certain cutoff or an age at which it will no longer be helpful at all, but I think the earlier we use it, the better. The beauty of SLOS in part is that I think many of us would agree that a big problem here is myelin in the brain. Myelin is the insulator of nerves, and it's made up of a lot of cholesterol. I think a lot of us think that myelin is abnormal in SLOS. If we could increase the cholesterol in the brain, it would become more normal. Well, myelination in the brain is occurring all the time, and significantly up to age 20 or 25 years of age, we're still making more myelin in the brain. And so I think there's an opportunity to, fix that at least through early adulthood and make some improvements there.
My daughter is 28 and is just as recently as 3 weeks ago doing new things.
Yeah. Excellent case in point. Yep. So I think we could see some improvements in adults, but I don't think the improvements would likely be as significant in younger children. But who knows? Mm-hmm.
Other questions?
To that, I have my daughter is 47, and I've had very little to no interaction with anybody with SLO, period, through those years. In the beginning, it seemed like, and we come from the University of Iowa, I come from Iowa, it seemed to be that they were interested in her, and there were some case studies. As she's gotten older, there seemed to be less interest. The older she got, the less interested anybody was to do a case study or to use her, my knowledge and her, changes and aging to help younger people. When she was around 23, I believe, I did find out about the cholesterol that was available, and I was told that she's a full-blown case, so it probably wouldn't make any difference. She does eat 2 eggs a day. I believe there's more than that. I'm very curious to find out why. But at different times, I've reached out, as I said, when things have come up, and I've had very little response back from the medical profession in regards to her. Last year, in fact, it's interesting, Dr. Opitz I met in Madison, Wisconsin when she was very young, and he told me, I remember, that that they do not get cancer, and neither do their family members. Last year, she was diagnosed with breast cancer. It's not genetically inherited. Although, again, that was 47 years ago, and maybe then they didn't believe that, but I was curious about that as well. But that's when I reached out again and tried to get a hold of somebody, Dr. Kelly, who's no longer, I guess, active, if they could help me somehow with a connection with research with that. I got no response back from anybody, and I'm just curious as to why, if that's the case, if other parents have found the same thing. And by the way, too, that's my daughter, Carrie. When I did start giving her eggs, though, my son and my other family members had said they thought she was smarter. She responded quicker, so I believe there is a difference in the eggs and the cholesterol level. So I guess just a response back from that. If you feel is that true with all diseases? As they get older, there seems to be less interest?
Yeah, really good questions, tough questions. First of all, the cholesterol supplementation, I still generally recommend it in my patients, regardless of age. It's a cholesterol deficiency syndrome. Individuals have low cholesterol. We can easily give them something that we know is safe that will increase their cholesterol. So I generally recommend it, even though we haven't proven that it's helpful. Anecdotally, I hear the same stories you're telling me from a lot of families. I observe it in some families where some of the kids or some of the adults seem to be better when they take cholesterol. So that's one of the reasons why I generally recommend it. I don't fault families who don't want to do it. It's up to them, but I usually generally recommend it. Adults with SLOS, and is there less interest? I think a number of us researchers, we generally come from a pediatrics background. I think most of us who are here today, the clinicians and the researchers, are geneticists, so we're very comfortable seeing and taking care of adults as well. Probably 20% of my patients that I see are adults. There should be more research on adults with SLOS. I absolutely agree with you. I think there will be in the future, and I think I speak on behalf of my colleagues. We are interested in adults with SLOS. There is still a lot to be learned, and I think a lot of my colleagues do have the ability to see patients who are adults in their clinics. So I think, let's keep in touch through the foundation. Gretchen has a list of us clinicians who are currently seeing children and adults with SLOS, and we would be happy to see some of these people. But there's not currently a study specifically looking at adults with SLOS.
Okay. Again, the cancer situation, I was going to
Yeah.
speak with you.
Dr. Elias can comment on that.
That was a desperation in a moment, because I clearly, when they told me that, I remember Dr. Opitz saying they don't get cancer.
It's one of the reasons why
And I
we
don't have
need
a
more
history.
research, because we can't just say things like that without doing the research and
Right.
knowing the answer.
Right. And if she could be helpful in any way to younger kids.
Dr. Steiner and I were just talking this morning. I asked him if he had seen any patients with cancer, because one of my patients in his early 20s was just diagnosed with metastatic testicular cancer. And it took longer, I think a little longer, for his diagnosis to be made, because he was living in a group home, and it was just that his mom came to visit him that she noticed a testicular mass. And thank God it was summertime and he was wearing shorts. That's how she noticed. But it's not something that's reported in patients with SLO to be certainly of increased incidence, and I think people get certain cancers at certain ages for lots of other reasons. We don't know all the genes that cause breast cancer, for example, and is it just true and unrelated, or is it something to do with SLO? In my experience, this patient of mine was my very first patient to have cancer. I'm currently following a 64-year-old, a 53-year-old, many patients in their 20s. So I do see adults as well as children, even though I'm a pediatrician. My training has been as a pediatrician, is with younger children, but as a geneticist, I don't care if you're 102.
Well, and hers is triple negative. And again, we did genetic studies, and it's not inherited. So it must have been, if it's not SLO, somehow it's just kind of a fluke that happened. And as far as finding it, she was in a group home 27 years, and in the discharge the last 2 years, there were lots of incidents. And so I was over there quite often, and the more often I was there, the more I saw. So she was discharged, and with a 30-day notice, and it was because they said of chronic dissatisfaction on my part, even though I had pictures and proof of things. But I believe, and my son had said things happen for a reason, I believe that if she were there, they would have not found the lump, because I've always been extremely in tune whether she's been there or not, and on stuff immediately, which is why one of the reasons she's 47. But I don't think that they would have found it, and we found it with 1A, I believe, 1B with triple negative. So
That's good.
anyway,
Good prognosis.
So I just wanted to respond to your comment about using eggs. So Dr. Steiner and I have known each other for many, many years, and I think we're friends and respect each other. I agree with him that cholesterol is not a cure for Smith-Lemli-Opitz, but I feel fairly strongly, and with my experience in the past 26 years, and caring for many, many patients that giving cholesterol to a person, especially if your cholesterol level is really low, does have beneficial medical effects in a number of areas. Helps the GI tract work a little bit better, and so it helps with some of the feeding issues. My colleague and I, Mira Irons, invented a cholesterol medicine that's the equivalent of 5 eggs per teaspoon, and it's very easy to give. I'm still using that. And I had an FDA IND number initially, and I no longer have to use it. Have permission from the FDA to use it as a medicine now, because it's been very safe and helpful. So a medicine may not be a cure, a proven cure for something, but that doesn't mean it has no benefit.
I didn't know, was there certain, how many eggs? Does that make a difference on how many eggs or any?
There is the body will only absorb so much cholesterol, so
Okay.
once you get to, as an adult, 3 or 4 eggs,
Okay.
you probably can't go beyond that, or it'll just come out in the stool.
And it's the whites and the yolk? Or
The
one?
yolk is what's cholesterol.
Okay, okay. Don't want to take any more time. Thank you very much.
Thank you.
JB Roulet, and I just wanted to follow up very briefly on the cancer and steroid. From a research standpoint, there's a lot of reports linking oxysterol with cancer, not in the context of SLOS, but those oxysterol are very DNA damaging, and they promote the growth of cancer cells. So in the context of SLOS, and that's purely speculative here, I think we will hear about prevention of antioxidant and prevention of oxidative stress with vitamins and Aquadex and other things, and that might be potentially a way of preventing whatever increase in cancer risk there might be in SLOS. So I just wanted to point this out.